let's see how we will do immunostaining when we have our sample, for example our cells, on cover slips. First of all, we will create a humidity chamber. So this kind of culture plates are coming quite handy, and then you can see I covered one with some aluminium foil to create also a dark chamber. Then we are taking a piece of paraffin, we are placing it here in the middle, and then we will just draw here at the corners a little bit. Okay, and we can also now write here, for example, the samples we have, control, treatment 1, treatment 2, T3, treatment 4, etc., etc. Then we take a bunch of papers, which we will then soak with water, quite a lot, not dripping, but they should contain a lot of water, and we'll place them around the area of this tray. So I just soak them. You see they are not dripping, but they are quite wet. And just go and grab some more paper and soak it with water. So everywhere where I want to have my cover slips, I will first add some PBS, a drop or so. For example, I want to have a cover slip here, a cover slip there, and a cover slip here. So in this 20 wire plate I have some cover slips with cells on them. I already performed fixation, permeabilization, and even blocking in this in the 20 well plate. Now of course theoretically you could also continue and add the primary antibody and then the secondary, etc., also here, but the volume that you would need would be quite high. You would need at least 200 250 microliter to be sure that the surface of the cover slip is properly covered. And that's quite a lot of antibody to use up. So what we will do now, we will basically lift up the cover slips with our cells on, and we'll place them in this humidity chamber. And to do that we need a needle with a slightly bent tip, and we will need a pincet. So I just lift the cover slip and then I grab it with a pincet. I put it here in the first place with the PBS, and I go to the second one, and I do the same. I go to the third one. Sometimes you need to be a little bit patient, but and so on. So I added the PBS here because if you have a lot of cover slip, you do not want them to dry out by the time you are finished of lifting them out from these wells. That's not a good thing. So now I would add the primary antibody, which I already prepared here in this Appendor tube. So first I will remove the PBS that I have there. And I will grab another pipette with 80 microliter, and I will just add it on top of my cover slip by the edge, not in the middle but at the edge, and you can see it covers quite nicely the whole surface. Probably it will even work with less, maybe 60-70 microliter, but don't overdo it because you still will have to incubate it for overnight, so you don't want it to dry out. That we added our primary antibody to all our cover slips, all we have to do is to cover the chamber, and now you can place it at 4 degrees for an overnight incubation.